Hello and welcome back to SPARCS 18. It is my absolute pleasure to be here to introduce you to our next speaker. We are continuing to explore the themes of behavioural genetics and looking at the evolution and artificial selection that relates to dogs and our interrelations with them. Our next speaker, Dr. Kristopher Irizarry, received his PhD from the Department of Biochemistry and Molecular Biology at UCLA, where he developed computational methods to identify functionally relevant genetic variations in the human genome. After completing his doctoral work, he accepted a postdoctoral fellowship and focused on identifying specific gene variants that were associated with the susceptibility to depression. His work led to the identification of specific genomic regions that predicted clinical patterns of anxiety and depression. Chris currently holds a faculty position at the College of Veterinary Medicine at the Western University of Health Sciences in California, where he is the director of the Applied Genomics Center. In 2009, his research expanded when he and his colleagues were awarded funding to sequence the genome of the endangered snow leopard. Chris studies comparative mammalian genomics with an emphasis on decoding the behavioural and anatomical information encoded in animal genomes. Remember, you can participate by sending comments and questions through Twitter to hashtag SPARCS, SPARCS18, all the way through the talk, and we will relay any questions to Chris in the question and answer session straight after his presentation. Please join me in welcoming Chris.
Hi. I'm going to start my session with an introduction to my background and a little bit of an overview of genetics and how it relates to organism development and organismal biology. And I wanted to go over my background because it's amusing to me, because when I first started out in science, you know, the idea was like, oh, you're just going to be a footnote in the annals of some kind of journal somewhere. And that's kind of how it ended up, and I'm thrilled about that, and I'll show you how. But genetics has been something that's been going on for quite a long time, and early, like in 1860s, Mendel started working with peas, and then a lot of years went by, and at the beginning of the 90s, we started getting into the genomics era, and in the beginning of the 90s, genomes were starting to get sequenced, like bacterial genomes. And then around 97, 98, 99, a big push came, and the human genome was, the draft sequence was released in 2001. And that was marked with these two journals, the Science Journal and the Nature Journal. And these were complete issues dedicated to the human genome. And as was already mentioned, my background, I did my bachelor's degree at Rensselaer Polytechnic Institute in Troy, New York. And I did my PhD at UCLA, and then I did a postdoctoral fellowship at the Neuropsychiatric Institute, and currently I'm a professor of bioinformatics and genomics at Western University of Health Sciences. And my first paper that I got in graduate school was in 2000, and we were like one of the first groups to identify close to 50,000 genetic variants in the sequencing data from the human genome. And from that point, I was always intrigued with, like, what do these variants mean? You know, we all look different, we all have different features, and I'd always wondered, can we predict something of value about an organism or an organism's traits from that information? And then when the human genome paper came out, it was a really exciting time. And this isn't my work, but this is a paper that was in there in the same issue. It's called A Map of the Human Genome Sequence Variation Containing 1.42 million Single Nucleotide Polymorphisms. And what's amusing about that is that one of the references in that paper has my name in it. So I have like a footnote in the Journal for the Human Genome, and that's amused me ever since. But since then, I've done some other work. I've worked on some bioinformatics software development to try and integrate information from different databases so that we can make informed decisions about genomes and genome sequences. And you know, as I started developing a better understanding of how genes and genetic variants could be interpreted in the context of specific phenotypes, I had worked on putting together kind of like databases that would highlight genetic variants that were likely to influence specific traits like obesity. In some of my work, you know, complexity is a big part of genomics, and dealing with complexity is hard. And people don't realize it, but if you have a genome that has 25,000 genes in it, if you talk about any single gene, that there's 25 individual genes, but if you start talking about pairs or trios of genes, you start getting into astronomical numbers. It's kind of like the lottery. There might be a lot of game that you have to pick 6 numbers from like 1 to 60 or 1 to 70, but to win is really hard to get the exact 6. There's many, many possible combinations of 6 numbers. And that's like a reoccurring theme in genomics, is dealing with the complexity. And so, I've worked and come up with some algorithms to reduce the complexity to try and get at answers when answers seemed difficult to get. I'm very much interested in managing captive endangered species. I work on snow leopard, and that's a challenging problem because there's only about 150, 200 snow leopards in North America. And I know when some people see someone breeding dogs, like, that's a backyard breeder. Well, I guess the snow leopards are the definition of a backyard breeder, because you're trying to maintain the species for the foreseeable future from a population of a couple hundred animals. So you have a genetically bottlenecked population, and they have a lot of problems. Like sometimes they're born with ocular coloboma, which causes blindness. They have like 30% cub mortality. So I've been working on trying to find genes that are associated with these traits. And there's a philosophical, ethical issue about we don't want to breed anything out of the genome. So it's a dilemma of how to integrate genomics information, but that's like the perspective I'm coming from, trying to apply the science to a situation in which we can make life better for these endangered species. I'm intrigued by comparative biology, and I chose to go into veterinary medicine because there's a large number of species. So here are 2 papers I've published on pythons. One of them was associated with trying to identify genes that are associated with spermatogenesis and sperm production, because there's kind of a python problem in Florida, but there's also endangered species. So if we could better understand how they reproduce, maybe there's ways to intervene when you have invasive species that are overtaking niches that don't belong to them. And so, in that paper, I took a lot of genes that when you knock the gene out in mice, and you have a defect in how sperm are formed or the shape of the sperm or the motility, I found those genes, and then I mapped those genes to the python genome. And so I took the knowledge from one species and applied it to another. That's called comparative genomics. And that allowed me to kind of get a handle on this, even though no one's really studied reproduction in these species. And I've also interested in the coat color in reptiles is like the pattern on the scales, the colors. And another paper I did looked at that. It looked at transcription factors. And so genes are, we hear the word gene, but genes are turned on by other factors called transcription factors. And then there's locations in the genome where transcription factors can bind up like next to a gene. And when enough transcription factors bind, the gene is produced and turned on. So, I did that in the pythons. I also did some other work on cats. I was in a collaboration with some people, and they had sequenced just over 1,000 genes in the cat, and they wanted to know what they did. And I did a comparative genomics map between the cat genes and human disease and nutrition. And we figured out that a lot of the genes were involved in biochemical pathways that were associated with wellness in cats, and that information was used to help improve feline nutritional products. Naked mole rat's an interesting rodent. It lives like 27 years and can't get cancer. So I did a comparative study between the naked mole rat and dog, human, and mouse, and I asked, what are all the genes that cause cancer? And if I find the dog version, the cat version, and the mouse version in animals that get cancer, and then I find the version in the naked mole rat, which of the genes are most similar between naked mole rat and the species that get cancer, and which ones are most different between the naked mole rat and the species that get cancer? And I hypothesized that the ones that were most different, maybe that was a clue as to some of the genes that were contributing to cancer in the species that get cancer compared to the ones that don't. And so from my perspective, I think that genomics is like a puzzle, and if you could find like nice ways of getting contrast, putting 2 things together and seeing what comes out of it, that can give you some insight into how things are encoded in the genome. I did some work on chickens because immunology pays the bills in the laboratory. You can get funding for infectious disease. So we had 2 lines of chickens. One line of chicken had a very robust immune response, and they've known about this for like 20 years. It has a region of the B locus, or MHC, the major histocompatibility locus, and there's genetic variants in that region in one line of chicken, and it can mount a robust immune response, and then in the other line of chicken, it cannot. And people have been wondering what's the difference between them. And we didn't sequence the genome of the chicken, but we instead took macrophages and we let the macrophages develop in culture, and then we sequenced the genes that were on in the macrophages at different stages of immune stimulation between the two lines. And I had almost 950 million sequencing reads, so about 1 billion data points. And I systematically went across. It took a long time to do, but I figured out that the genes were turning on in different temporal patterns between the two. They mostly had the same genes, but you could think of it like you have a car manufacturing plant, and in the robust immune line of chickens, when the wheels are ready to get put on, so were the axles, and so were the lug nuts, and so were the tires. But in the other one, it would turn on the tires, but the axle wouldn't even be produced, so it was producing like incomplete components for the immune function to work. That was kind of exciting. And then there was another follow-up paper to that as well, looking at microRNAs, which are small molecules that can inhibit other genes by binding to the mRNA and preventing it from being translated. Some of my most exciting work that I'm really happy with is the work that I've done with Dr. Voith. Dr. Voith is also a faculty member at Western University in the College of Vet Medicine. She's one of the founding animal behaviorists in the field of veterinary medicine. And she and I had worked for a while on seeing, people have thought for many decades that they can visually determine what breeds make up a mixed breed dog. And what we've learned is that you can't really do that. And if we compared breed test results to visual identification, you don't have overlap. And even when hundreds of people agree that is a lab mix, even though a large number of people agree, the genetics can say no, it's not. So there's quite a bit of implications for how we identify dogs and the attributes that we assign to dogs and what we think we know about breed. And so I was really, really thrilled to be able to work with Dr. Voith on that. That was really cool. So, I want to give you my perspective on life. I look at things in a very global perspective. And, life has been on this planet for billions of years, and I don't think of it as we're all separate. I think of it as one continuous flow of cell division that's just kind of radiated out. And from that perspective, I think of biology of scale. We talk about gestation, and you can go from a single cell to a complete organism for humans in 9 months. And in other species, maybe 20 days, 21 days, or 30 days for some rodents, and dogs are a little longer. And then you think of evolution, and that's hundreds of millions of years. So time is a very interesting concept in genetics, and we use time as a measuring stick to make genetic inferences. So our measuring stick can go from days to weeks to months, all the way out to tens of thousands of years, or hundreds of thousands, or millions of years, and even billions. So in this graphic, I just want to let you know I take all my images from Wikimedia. They're open source public images, and I cite all the images. So whoever created them, I'm very thankful for, and that's why I have these large links underneath my images. But in this image here, this is what I think. You have a constant flow of cell division. And if you think of gestation, I want you to think of gestation for a bit. You have an embryo, you have a developing baby. In your mind, that's like one entity, right? It's composed of cells, but it's like one entity. And then the cells divide and you get a heart and lungs and a brain and eyes. But that's one process. And so if you step back and open your mind a bit, you think about life. Life is the same thing. It could just be one massive gestation of life just flowing on Earth for billions of years. And from that perspective, genetics gets to be really interesting. So if you look at everything we know about the universe, I'm just going bigger now. 13.7 billion years is the estimated age of the universe. And right here is kind of 3.5, 3.7 billion years ago at this purple arrow is when life started on this planet. And there's publications that talk about different aspects of evolution. But just to put it in perspective, 5 billion, 4 billion years, 3, we start to get bacteria and some multicellular organisms about 1 billion years ago, 800 million years ago. And then about 500 million years ago, fish emerged. And then we get, some other animals like reptiles, amphibians. And you come down here, we get 10 million years ago, we start getting more towards human line. And the point is that this is a constant flow. If you think about it, no organism that's alive today, well, now it's kind of possible, right? Because we have genomic technology, but every living organism has come from another living organism, and you could trace your ancestry back billions and billions of years. So one thing we talk about in genomics is the last common ancestor. So we could say, we're human, and we have dogs, we could ask what's the last common ancestor between humans and dogs, and so that might be some many millions of years back, 50, 60, 100 million years. But that concept is kind of important to keep in your mind, that there was once an organism that had descendants that became humans and became dogs. And then the genetic variation and the mutations that occurred over time are what gave rise to the different morphological features that we call phenotypes and the different patterns of behavior that the different organisms display in their particular environments. So you could find in literature, like, this is a gene called heat shock protein 90. It doesn't matter. It's just an example to show you that they're saying 1 billion years of evolution in this paper, meaning that gene has been in organisms for 1 billion years, and we have that gene, or paralogs, which are related genes, to it in us. And then there's another paper here that talks about finding evidence of life from 3.7 billion years ago. So life's been going on for a long time, and all of that life is based on DNA, and all the permutations that the genome can incur during evolution are what give rise to all the forms we see today. So now I'd like to give you like a brief introduction to genetics, and I'm just going to make this simple, and I'm also going to say that this is 80% correct and 20% wrong. And if the 20% that's wrong starts impacting you, then you're in a position where you need to get more information in this talk. Frequently, when people give talks publicly to mixed audiences, sometimes people ridicule them or criticize them for not being technically accurate. There's no point in raising all of the exceptions. I'm just trying to get everyone on the same page, so I'm just going to very much simplify this so it makes sense. And it may not be technically accurate, but it's good enough for you to understand the second half of my talk that's going to come next. So we have chromosomes that are inside the nucleus, and a genome is the set of chromosomes. And what you see, like these X's, are really two sister chromatids. The chromosomes are linear molecules, but they're in pairs. We're diploid, means two copies. And so that X is two copies of a chromosome, and that's why you could be big A, or big A, little a, or little a on a gene on one of these chromosomes, because there's two copies of it. One came from mom and one came from dad. When you look at the DNA, it's made up of bases, and these are the 4 letters of the genetic code. But if you think of Morse code just has dots and dashes. It's two letters, but you can encode the whole human alphabet with the dots and dashes by using more than just one. You could use a variety of dots and dashes together to get more complex representations. So 2 things I want you to know about DNA are that there's bases, there's 4 of them, and the DNA molecule is 2 strands, and the bases are always paired together. They're like little magnets that you ever play with magnets, and they like stick together. That's how the strands of DNA are. So C and G are going to bind to each other, and A and T are going to bind to each other, and that's called base pairing. So the structure of DNA, which was figured out by Watson and Crick in 1953, at the end of their paper, they say something very vague. It's like, oh, the structure of DNA suggests a mechanism for replication. It does. Since we know that the bases base pair, with A always pairing with T and G always pairing with C, the process of cell division and cell duplication and chromosome duplication occurs by DNA unraveling, and then each strand serves as a template for the assembly of free nucleotides to come in, and since G binds to C and T binds to A, the appropriate base pair letters line up, and then an enzyme connects them, and then you have a new strand of DNA. So DNA is self-replicating because of that process and those features biochemically. So now I want to talk a little bit about cell division, just because it's important to understand, but then we're going to get into some of the complexities of how cells differentiate. So we have stem cells, and then we have cells that are differentiated into like muscle or neuron. So cell division occurs when signals in the cell or signals external promote the cell to start duplicating its genetic material and to start taking in molecules to make new organelles and the internal components of the cell. And that occurs, and then eventually all of those things line up. And then the cell, there's genes that turn on that create what I consider to be like the ropes on a massive ship, like something that came over to America in the 1600s that held all of the, held the ship together. So those ropes come and grab the chromosomes, and then they literally pull apart, and half of the chromosomes, go to one side, and like one copy goes to one side, one copy goes to the other, and then the cell kind of pinches off. If you imagine like taking a balloon and putting a piece of dental floss around it till it gets really tight, and then it kind of pops in half and you have two cells. So that's cell division. And when that happens, sometimes you can have asymmetrically some genes that were on in the parental cell, you could have those gene products only in one of the daughter cells and in the other none. Or you could have disproportionate concentrations in them. That's one way you get asymmetry and differentiation. And like the most powerful stem cell is the fertilized egg, right? Sperm and egg join, and that's going to become a whole organism. It has to make cells. But there's different types of cells in a developing embryo, like endoderm, mesoderm, and ectoderm. And they give rise to different things. Like endoderm could give rise to like lung and pancreas. Mesoderm could give rise to muscle and blood cells. And then ectoderm gives rise to brain and skin and pigment. So this is kind of a karyotype of humans, and I'm just showing you that the way we talk about chromosomes is we number them. And we have two types of chromosomes. We have chromosomes that are not involved in sex determination, and those are called autosomes, like automobile, but autosomes. And they're generally numbered. The lowest number is the largest chromosome, and the higher the numbers go, the smaller the chromosomes are. So humans have 24 different chromosomes, but we say there's 23 pairs, 22 pairs of autosomes, and then a pair of sex chromosomes. In women, it's two X's. In men, it's an X and a Y, but Y and X are different. The interesting thing is that the human genome is about 3 billion letters. That's not the diploid genome, 2 copies, that's just one, 3 billion letters. And according to that science paper that came out, about 25% of, if you stretch that 3 billion letters of all the chromosomes out, and you just closed your eyes and touched any part of a chromosome, 25% of the time you will be in a gene, 75% you'll be between the genes. And within the genes, the 25%, over the whole genome, 1% actually codes for the protein products of the genome. So 99% of the genome is not coding. And it amuses me, people have called that junk DNA. I call it idiot DNA because only idiots would think that 99% of a genome that sustained itself for millions of years is actually junk. And I don't mean to offend anybody, it's just like my little joke, don't take it personally if that was you the one that called it junk DNA. You were very wise because I didn't even know there was DNA when you figured that out. All right, so one other thing I'll say is that, it's debated how many genes there are in a genome, and you start counting, it gets weird. We used to say one gene, one protein, then we realized that genes can splice together. Like, if you took a book and you Xeroxed it and then ripped out 5 paragraphs from chapter 4, that's a different version of the book. Genes do the same thing, it's called splicing. But if we just say there's 25,000 genes in the genome and you have 20 chromosomes, so on average, right, there's about 1,000 genes per chromosome. But because chromosome 1 is larger than chromosome 22, maybe there's 2,000 on this one, and maybe there's 600 or 500 on that one. You see what I'm saying? So the idea is that the genes are distributed over the chromosomes, and if we know which genes are responsible for which traits, we can better understand how biology works, and we can understand how genetics plays into traits. So I was telling you, when we zoom into a chromosome, we have a gene, and the exon is the part that's expressed in protein, and the introns are the interrupting pieces. And then there's also a promoter, which are the control regions for a gene. And I'm going to try to explain the combinatorial regulation of gene expression in about 2 sentences, and it works like this. There are proteins that recognize certain DNA sequences. And so you might have a sequence that's like GGAATT, a certain transcription factor can bind wherever that sequence is. It may just so happen that evolutionarily, that sequence only occurs in genes related to mount an immune response. And so when your body says we need an immune response, it turns on a transcription factor, it goes in the genome and binds everywhere where there's a GGAACC, for example, and that happens to be the 160 genes that the macrophage needs to turn on to fight an infection, for example. So the genetic code is really cool, and I told you it's like Morse code. So when we look at DNA, the way I look at it is I see these letters, but there's this chart. And so if we see here this ATG at the beginning of the bottom of this blue strand on the top where it says template strand, I'm on the bottom strand ATG. We go A from the inner circle of the wheel, A, T, and G, and that codes for methionine. Methionine is an amino acid. The amino acids are like little crosses or plus signs that all 3 parts of the plus sign are identical, but 1 part is variable. And the way to think about them is some of the variable parts, so different amino acids have different properties. Some are like water, some are like butter, some are positively charged, and some are negatively charged. So when a gene is turned on, it produces an mRNA that is read by machinery in the cell, and then the proteins you eat in your diet are broken down and made available to your cells, and then they align, and then your body connects them together like a paragraph in a book. So when I look at a gene, it looks a lot like a paragraph in a novel. You know, you might have like 15 sentences, maybe it's 375 letters, maybe it's 800 letters. There are 20 amino acids, there's 26 letters in the English alphabet. So it's a really good analogy of thinking of like a protein sequence like a paragraph or two paragraphs. So remember, we have the chromosomes in the nucleus, and then different genes on the chromosomes can make different proteins, and those proteins can assemble to make larger machines or machinery. And the way I think about proteins is that they serve just a handful of roles. But if you needed to understand what is a protein, if you think of your car, a protein is everything that makes your car, from the bolts to the components in the engine, the tires, the wheels, all of that. So the way proteins function is that they make a very distinct shape. And if you think of a Christmas tree and you think of the Christmas lights that you hang on the tree provides support for some parts of the lights. And then where there's gaps between the limbs of the Christmas tree, gravity's pulling down and you get like a loop falling down on the Christmas lights. Some parts of a protein are positively charged and some parts are negative. So if there's a positive and negative, they'll attract, so it'll pull it together. And if there are two positives, they'll repel each other like magnets that repel. So what ends up happening is you get a very distinct three-dimensional structure. And I want you to think of tools like a socket set or a wrench or a screwdriver. The reason why a Phillips head screwdriver takes out a Phillips head screw is because it has the right shape to fit. And that complementarity is a big theme in biology. So again, just kind of giving you a feel for like how we think about genes and what they do. And then you might find that, you know, the development of eyes might be dependent upon a certain transcription factor turning on genes that make the eye form. And I think of genetic signaling as a lot like dominoes. You line up, you might have like 10 or 15 dominoes in a row. You push the first domino, that's the first gene, and then it signals to another gene, the whole thing falls over. If you have a mutation in a component, it's like pulling out one of the dominoes. Now one domino falls and there's no space to hit a domino in between, so the domino chain fails and you don't get the whole domino effect to fall down. That's where mutations come in, and that's where you can get disruption of certain processes. So from protein structure, I told you that the amino acids are just a linear string, and they have like 3 levels of organization. They could be strands or helices, kind of like a spiral staircase. And then that's like local regions. So maybe this small region might be a helix, then a strand, then 2 helices, then a strand, a strand, a strand, a helix, a strand. And then those things associate in higher order. And then you can get what's called like a tertiary protein structure. And enzymes are proteins. And it might be that in order for an enzymatic reaction to occur, like let's say that you wanted to break down starch, you have to cleave a bond. Starch is many sugar molecules connected together. And so maybe you have an enzyme like amylase that is able to break down starch because it has the shape that's complementary to the bonds of the starch molecules, the individual sugars, and it might have the proper electric charges so that when it's there, it grabs an electron, it breaks a covalent bond, and then the molecule of sugar is released from starch. That's what we do when we need to utilize energy and we've been fasting. Our body breaks down starch. So zooming out now, we got chromosomes and we got genes on our chromosome. And now here's an example of kind of scale. Like here's the chromosome that you see. That's 1/2 of the X that I was talking about, the chromatids. And genes have very unique locations in the genome. So in a species like dog, if we said the dopamine 4 receptor, DRD4, is located at a specific chromosome at a certain percent from the bottom going up, maybe it's like 30% up from the bottom, then in every dog, if you had this genome and you go to that address, you will find the dopamine DRD4 gene. So when people say they've mapped a gene, they know where it is, and that gene can get turned into a protein, and then it can go into the cell to be a receptor that can sit there like a keyhole. So when the neurotransmitter dopamine is released, the cell that hears the dopamine signal can respond to it. And I'm just showing you scale again. Like this might be a little piece of a membrane of a cell, and you can see that there's proteins in the membrane, and that's just like the relationship there. So neurobiology is something that we have to talk about a little bit if we're going to talk about the biology and the neurobiology of the human-animal bond. I think the easiest way to think about neurobiology is you can think of neurons as they forward texts all the time. So a neuron fires and it like quickly texts his buddy, and that text goes down and it spits out a neurotransmitter, and the only way for that signal to be propagated is the neuron that's connected to it at the bottom has to be listening with the appropriate receptor for the neurotransmitter that's released. The neurotransmitters themselves are built from things that we eat in our diet, and then there are other genes in the appropriate cells that will break down certain things and build up other molecules. It's a multi-step process, and finally they'll say, hey, I built up a bunch of dopamine, and that'll be stored in these synaptic vesicles, and then when the cell fires, it will release dopamine, and then there's other genes that break down dopamine, like COMT, catechol-methyltransferase. And so it's a tight balance between producing these signals and making these signals go away. I mean, if you wanted to text somebody that you're home and you type home and they get the message home for the rest of their life, you don't really have any communication capability. So the signal has to be temporally limited so that it's on and then it's off, because that's what gives it the specificity and meaning. So any gene that gets perturbed that can change the amount of neurotransmitters that are produced, how easily they're released given an input signal, and where the dopamine receptor on the other cell is located, and how many receptors are present, and what the amino acids are that are in the dopamine receptor that have to recognize the dopamine, and then the amino acids inside that have to knock down the other dominoes. These are all places where you can get a mutation that could change an amino acid and change the way that signal is transduced. Now, one thing I'd like to say, I'm just going to go back a little bit to show you this genetic code. Here's something that's interesting. So let's say that we get a mutation right here in this G, ATG. Well, if we have ATG, right? ATG is methionine. If I change that G to an A, now it's no longer methionine, it's isoleucine. But let's say it was normally an isoleucine, let's say it was normally an ATA for isoleucine, and then a mutation at the third position changes that last A to a C, it's not going to change the amino acid. The genetic code is 4 bases, A, T, G, and C. The codon code, or the code for amino acids, is 3 letters at a time. So if you say 4 times 4 is 16 times 4 is 64, there are 64 codons. But for leucine, for example, anything that's a C and a T, no matter what the third letter is, it'll code for a leucine. But in other cases, you can have, like, for glutamic acid or aspartic acid, GAT is aspartic acid, but GGT is glycine. So it depends, like, you have to translate it. So what I'm saying is that when you get a mutation, it may or may not make a functional change in a protein. And remember, the protein coding regions of the genome are only 1% of the 3 billion letters for the human, and the canine genome is about 2.3, 4, I don't know, less than a human, but more than 2 billion. So the point is, just because there's a genetic difference doesn't even mean it's going to have a phenotypic consequence. So that's part of figuring out. Then you got to figure out what gene it's in. Then you got to figure out what that gene does. So from a neurobiological perspective, you can have genetic variants in the DRD receptor, and they may be of no consequence. Or you could have, amino acid changes aren't the only way that you could change it. There's molecules called tRNAs that hold the amino acids when the cell is assembling the protein. And there's different tRNA for each version of the codon that codes for, like, a glycine. So the concentration of tRNAs in the cell can vary between the different codes that code for the same amino acid, and some might be very prevalent and some might be limited. So depending on which code you have for the glycine, you may be more likely to be able to throw a glycine in that protein, or you may not. So it's complicated. But the point is that just because there's a variant doesn't mean you have a change. So from neurons, the brain, briefly I just want to mention that in humans, brain and communication are kind of starting to be understood. There's still a lot we don't know. But we know, like, the frontal cortex is involved in kind of more complex planning and organizational thinking and thoughts. And dogs have a cortex too, it's just not as big as ours. One of my favorite things is when people say, oh, you're anthropomorphizing. I'm like, no, that dog looks hungry. They're like, I think you're anthropomorphizing. I'm like, no, look at him, he's standing there, he hasn't eaten in 2 days. It's like, some people are really incredulous. They think that humans are unique to certain experiences, emotions, but comparative anatomy and comparative biology tell us that if the structure is conserved, then the function may be conserved. So I feel like because there's a lot of relationships between brain structure and between mammals, I think there's a lot more that mammals share in common between emotions and feeling and thought than we originally believed. And interestingly, they're starting to now develop technology to do live, awake, functional MRIs on dogs. You train the dog to be very still. And you have to train the dog because you have to keep the dog still. And now they're starting to do things like talk to the dog while it's in the fMRI machine. And whoa, the same part of the brain lights up in the dog that lights up in a human when you ask them a question or you say the name. And that's just more evidence that dogs are more similar to us than we initially thought. So this is what MRI scans of the brain look like. And this is a dog reading. All right, a little bit about developmental biology. You start with a single cell, and then it divides, and it keeps dividing, and then it becomes something called a blastula, which is like a ball with a hollow interior. And then it goes through gastrulation, where there's kind of like taking a balloon and pushing a pitcher inside, putting your finger inside and pushing it. That becomes like the gastrointestinal tract, if you will. I'm exaggerating. But the idea is that that's how you start getting more complexity out of it. And genes have certain promoter regions or control regions that are linked with the cells and the tissues and the organs and the functions they play. And so things that are going to affect neurons, we can start to decode by understanding neural-specific promoters or neural-specific enhancers, which are the control regions. And we also know if it's a neurotransmitter receptor, it's probably expressed in the brain if it's a neurotransmitter receptor, but it could also be expressed elsewhere as well. Like biology is full of surprises. But the point is, I just want you to realize that, we can track things. People have done studies where they've looked at embryology, and they used to do studies where they would cut embryos in half and see how they develop, and they would take a tissue that was taken at a certain stage in development and hold it against an embryo that was in an early stage, and they would get development of a later stage in it, so they'd know that there's a protein that's secreted from the graft tissue onto the host tissue. And so, literally, probably thousands and thousands and thousands of hours of people studying this. We figured out a lot about developmental biology and about genetics. And so now we can start to understand some of the things that are involved in creating the body system. So this is an example I like a lot. So there's a gene called myostatin. And myostatin, you might as well call it myostoppin. Myo means muscle and stop means inhibit. Myostatin is stopping muscle. So normally this gene is involved in limiting muscle production. Now, we talk about dogs and selection. In whippets, racing is the application that they're bred for in the working capacity. And people were breeding whippets, and they were getting some fast dogs. They had some lean, muscular dogs, and they were pretty fast. And it turned out that they had a mutation in myostatin. And that mutation caused myostatin to stop working. And so if you were heterozygote, you'd have this lean, muscular physique, and you'd run pretty fast. But if you cross those fast dogs with other fast dogs that had the same muscular physique, you'd end up with the recessive version, and you'd have two copies of this gene, and you'd have these very bully dogs that look like a bully whippet. It's not related to a pit bull. It's just they have no functional version of myostatin, so they have a lot of muscle. And just again, anatomically, we talk about head shape in dogs, brachiocephalic or mesocephalic or dolichocephalic. And here's just examples of them. And here's kind of like looking at the different views of the skulls. And people have teased apart some of the genes involved in that. So what I'd like to do in the last few minutes I have is just go over some important genetics concepts. I have about 8 concepts I want you to understand. I'm going to present them very simply, almost like just straight statements. So the first one is genetic variation. So if two people get their genome sequence, and you might find in a particular position there's one person has a C and one person has a T. That's the simplest type of genetic variant because these are called nucleotides, and because it's only a single nucleotide that differs between them, all these black letters on the top are the same and all these black letters on the bottom are the same as the ones on the top. This single nucleotide difference, or single nucleotide polymorphism, abbreviated SNP and pronounced SNP, a single SNP is the simplest type of genetic variation. And another important concept is genotype and phenotype. And genotype and environment contribute to phenotype. I want you to think of some animal that lives in a kind of snowy Arctic environment, but in the summer it's not white, but then the winter comes and it turns white. Why? So it's not like it was born with that. It just so happens that they had mutations in a large population that when certain seasonal changes occurred that affect hormones or affects other aspects of its biology, that would result in the hair being white because that saved it from predators. And what we got was a selection process. Or you could think about how flowers bloom in certain seasons. That's environmentally induced. And that's why genotype and environment together are responsible for phenotype. The next thing is genetic selection. If we have a SNP that differentiates between two forms or two phenotypes, if we just choose the top one and we just have a population that only has A where there's two letters, we'll end up with a subset of what used to be there, and that's kind of the idea behind artificial selection. One way to think about it is if you imagine a room of like 1,000 rabbits of all different colors, and then you just pick like one color of rabbit you really like, and you just take those and you just keep breeding those. Eventually, the offspring and the descendants of those rabbits will lack the alternate letters of the genome at those positions that gave rise to the colors you did not select. And so when you do a selection, genetics is a population thing, not an individual thing. And so when you have a population, you can choose from that population a subset, and after you cross a line, you're no longer going to be able to have the genetic variation that was in the parental population because you've selected a subset of that. All right, fourth, genetic association. One of the most powerful tools that's used in genomics is genetic association study. And the way that's done is you have two different traits of interest. Maybe you have like a cancer and not cancer, or you have any two phenotypes. And so you have a case control experimental design where you have 1,000 individuals with the trait, 1,000 individuals without the trait. So those are your controls. And then you try, super simplification, but you basically go through and try and find out if there's a genetic variant that splits with the split of the phenotype. And it's complicated because not all things are black and white. Sometimes you can have traits, so we call penetrance is like what percent of the time a trait is going to show itself if you have the gene for the trait and the allele for the trait. If it's 100% penetrant, then every time you have the gene, you're 100% of the time going to have the trait. But some diseases can be, you can hold off disease sometimes even if you carry a gene for it by like having a good diet or maybe taking better care of yourself and exercising. So even though you have the trait, even though you have the gene for the undesirable trait, you may not show the disease until much later. So that means it's not penetrant. I'm kind of making this up a bit, but the point is sometimes because of penetrance, people will put individuals who have the gene for the disease in the control group without knowing it because they don't have observable evidence of the disease. If you imagine if you do a study on cancer or something, you don't want to take a bunch of 6-month-old puppies because they haven't lived their life yet. You don't know if they're going to get cancer. So there's those things. But basically, there's flaws with the design, but there's also a lot of value in it. And so a lot of times you get like 5 or 6 regions of the genome that show like these are likely candidates for being involved in this trait. The next concept is gene expression. So as I told you, you have DNA. DNA opens up when there's a gene that's turned on. mRNA is made, and then that mRNA is turned into a protein. We call that gene expression, and experimentally it is very easy to measure gene expression because DNA base pairs. So if we purify RNA and if we know the genome, we could put all the sequences of all the genes on this little slide, if you will, and then we put different color fluorescence on the nucleotides, and then when they hybridize to the slide from base pairing, the mRNA is able to base pair with the DNA on the slide. Not exactly, you synthesize DNA from it, but I'm just simplifying this. You can say like between cancerous cells and healthy cells, you might find that each coordinate here, each row corresponds to a gene and each column corresponds to a condition, like healthy, healthy, no cancer, early cancer, later cancer. The point is that anything that's expressed in the healthy cells will be colored green, anything that's expressed in the cancerous cells will be colored red, and anything that's expressed in both will be colored yellow. So we can kind of tease apart where, depending on which samples you choose, we can go, oh, we know these 375 genes are only on in cancer, or we know they're only on in the part of the brain that's responsible for speech in humans, or we know they're only on in the amygdala. Reproduction is a super important point. Basically, sexual reproduction, not only do you get a copy of a chromosome from each parent, the chromosomes recombine. And what that means is if I have a chromosome from my dad and I have a chromosome from my mom for chromosome 1 in my cells. The chromosome 1 that I have, my maternal chromosome 1, is a single chromosome that's got pieces from my maternal grandmother and my maternal grandfather kind of stitched together. So when they say I got, and my brother has a different chromosome 1 that's maternal because he has different parts of that chromosome 1 that came from my maternal grandmother and my maternal grandfather. And what this allows us to do in artificial selection is you could take traits that are in different animals and it's possible to cross them and end up with a single animal that has both traits. That's basically the take-home message. And then we have inheritance. This is dominant and recessive, and it's kind of hard to understand, but just in a nutshell, what I'm going to say, I want you to think of a bicycle, and I want you to think of a bicycle with its tires. And I'm going to say that a smooth ride on a bicycle is recessive, and an uncomfortable, lumpy ride is dominant. You have 2 wheels on a bicycle. Only way for a smooth ride is you need 2 round wheels. So you have 2 copies of the round allele, no other allele, you have a smooth ride. If you have 1 wheel that's not inflated right, you got a bumpy ride. That's dominant. So bumpy is dominant and smooth is recessive. See how it's just this weird thing? It doesn't really make sense, but you could go, oh yeah, that's how it is in biology too. Kind of like it just is or it isn't. So last slide I want to talk about right now is genetic bottleneck. And what this is each of these colors represents a different allele in a population of wolves, okay? And we're talking about the domestication, artificial selection of dogs. And so someone grabbed some subset from this, and each color is represented equally at like 1/5 of the total population. But as you start selecting now, you get down to like an overrepresentation of the blue, and then from that population you could pull, you might end up with a massive overrepresentation of red and no yellow at all, and that might be domestic dog and that might be wolf. So you see all those colors in there? When we compare the genomes from a dog to the genomes of a wolf, you will say like, oh, the wolf has, in the population of wolves, there's many, many variants in this gene. And then you go in domestic dog, there's very few variants, regardless of breed. So we believe that's evidence that that was one of the traits we selected on because we removed the genetic variation that was affiliated with it in the population. And so that region that doesn't have variation in the domestic dog population is the region that may be responsible for some of the traits that were selected. And I'm going to stop there. And this is another example of genetic bottleneck, but that's an important point. And I think that's it. And when we pick up next, I think I'll go over these points again when we come back just to refresh your memory. And I'll just stop right here for now, though.